I am involved in stem cell research, and I'm involved in a complex program involving IFOM and other institutions. Stem cells are the only cells that can regenerate an organ. So essentially, when we look at an organ, the only possibility of regenerating real tissue comes from stem cells, so that the majority of the cells in the organ are differentiated cells which originate from the stem cell. As a matter of fact, in normal tissues, really what happens over time is that stem cells decrease in number, and this is one of the leading cause of aging in the mind of several people, because basically the stemness, the capability of regenerating, gets exhausted. Now, not surprisingly, in cancer, a very similar situation occurs. Cancers are basically in several ways, they become paired to organs which are just being formed with an aberrant plan, and so they are also fueled by stem cells. And so stem cells are the only subpopulation of cells through which cancer growth can be achieved and cancer can expand and regenerate. Now, there are important differences between normal and cancer stem cells. They are not completely clear, of course. That's the major focus of investigation now in the field. From the phenotypical point of view, really perhaps the major characteristic of cancer stem cell is that for a stem cell to become a cancer stem cell, or for a more differentiated cell to revert to the state of a cancer stem cell, what needs to happen is what is called epithelial to mesenchymal transition. Now, in the epithelial to mesenchymal transition, the cell acquires essentially migratory capability in addition to several other phenotypes. And this is one of the reasons why one of the leading hypotheses in the field is that stem cells are really the cells responsible for metastasis. So they become, the importance in cancer becomes twofold. They not only regenerate the cancer, but they also cause what is the most dramatic occurrence in cancer, which is metastatization. And there is another characteristic which is important in cancer that we should bear in mind. It is that stem cells spend most of their time in a quiescent state, which means they do not replicate. As many of the weapons, of the drugs that we have at our disposal in cancer interfere with the proliferation one way or the other, not all of them, but a good number of them, there is the legitimate suspicion that stem cells will evade therapy simply as a function of their state of quiescence. So in a nutshell, these are the major phenotypical differences, at least from if you look at the thing from the cancer point of view. It started a few years back. We were involved in a collaboration with Pier Giuseppe Pellici's group that led to two major avenues of investigation. On the one hand, we developed technologies to purify stem cells to near homogeneity. We use a very simple method. We use a vital dye that gets diluted when the cell divides and does not get diluted if the cell is quiescent. And in this way, the stem cell retains the dye and can be FACS sorted. And this applied to the old mammosphere assay, which has been a basic tool in the field to identify cells which were capable of proliferation in vitro and making colonies in vitro, led us to the possibility of purifying stem cells in a number sufficient to do molecular genetics and to do protein analysis and this kind of studies. What you have to bear in mind is that, say, in a normal mammary gland, in the normal condition, there will be one stem cell out of 40 or 50,000 cells, or maybe even less. And basically you cannot cultivate stem cell because as soon as you start cultivating them, they will differentiate into progenitors, and so you will lose the stem cell. So essentially the work here is repurifying every time that you have to do an experiment. And the yield is everything that counts. So with this methodology, we manage to get enough cells for experimental need, both from model system, and but mostly from patients, normal, counterpart, and tumor tissue. By applying this technology, we found out a number of things. We found out, for instance, that the critical determinant is p53. And the presence of p53 in the stem cell is really what determines the quiescent phenotype of the stem cell. And the mechanism is very interesting because when the stem cell first divides and gives rise to two different cells, one that retains the stem cell fate and one that becomes a progenitor, and I'm now talking about the normal condition, the activity of p53 is only retained in the stem cell. And so this is the way through which the stem cells, in our opinion, withdraws into quiescence. Now the question is why this happens. And here comes our historical expertise. Over the years, we have been very interested in the field of membrane trafficking, which is basically all the infrastructure that in the cell organizes logistics. So endocytosis, transport, all the vesicles and biomembrane that regulates cell transport. We discovered a protein, well actually we rediscovered a protein many years ago, which is called NAM, which is involved in these pathways, which have been known for a very long time to be a cell fate determinant. And these are studies that date now, perhaps 20 or 30 years, in which by using model system like Drosophila, they found proteins that did exactly the thing that I was telling you about before, which is they partition in one cell and not in the other when the stem cell divides originally. So the idea that we got there is, well, if a protein like NAM does this in a model system, perhaps it does this also in humans, and perhaps this is important for cancer. And indeed, we found that in mammary stem cells, NAM partitions asymmetrically between the stem cell and the progenitor, the daughter cell that does not retain the stem cell fate. And to put everything together in what was a real, I think, fundamental discovery, we found out that NAM regulates p53. So now the circle is closed. And actually NAM regulates p53 because it prevents its degradation. So when there is NAM, p53 is stabilized. When there is no NAM, p53 is lost. Now, to make the story even more interesting, what we were finding out at the same time is that the expression of NAM is frequently lost in human mammary cancers. So now the picture started to become coherent, to make sense. So when you lose NAM, you would lose the stabilization power over p53, so there would be no p53, and so now the stem cell can proliferate more, or perhaps acquire even a new kind of destiny, which would be compatible with that stem cell making the transition to a cancer stem cell. And so this is really what we are involved in right now, trying to understand how this mechanism can force the conversion from a normal stem cell to a cancer stem cell, and how to interfere with this mechanism. Because now the entire story is that you can take a NAM-negative tumor, a tumor in which you can actually test that NAM is not present, and perhaps revert it to a normal state by interfering downstream with p53. And there has been a lot of interest, of course, in p53 stabilizing molecule, so another field now can converge over these things, and possibly we might stratify, at least at the experimental level right now, the patient that would be eligible for this kind of therapy, or at least the cells that we would try in the lab to be eligible for this kind of therapy on the basis of the NAM status. Another interesting thing that we did, starting with our purification procedure, is that we could purify stem cells from the tumor and from the normal counterpart from the very same patient. And so for the first time, we could make expression profiles which would distinguish the cancer stem cell with respect to the normal stem cell in a completely isogenic pathway. And by doing that, we identified a series of specific stem cell markers. Now, this led to a number of discovery. One, which we published a few years back, is that tumors, the aggressiveness of tumors, of breast tumors, is a direct function of their content in stem cells. So tumors which have a lower number of stem cells are less aggressive, and these patients have a better prognosis than patients that have a higher number of stem cells. This is important, and we could do it only because we finally got specific markers, so we could actually stain in a stem cell specific way the tumor and actually count the cells in the tumor. And that led also to a distillate of these expression profiles, which was condensated in a signature, which we are now validating, and that is very promising in terms of patient stratification and eligibility for more or less aggressive chemotherapy in breast cancer. Well, basically, based on the signature that we have developed, we can assign a risk score based on an algorithm that takes into consideration the expression of every single gene and weights them on the basis of their impact in the whole signature. And so every patient will be assigned a risk score, which is a risk score of metastatic relapse and eventually unfavorable outcome. Well, I mean, the idea here is to use the signature in a way which is not different from the way in which other signatures have been used. We hope that our will do better, of course, but that's early to be said. So the general idea is to go into groups which are already existent in terms of treatment, like taking luminal tumors that receive certain kind of therapy and stratified within the luminal or within the luminal A and within the luminal B. So to use the signature as an additional tool to dig deeper into something that cannot be resolved right now. We know that in every subgroup of breast cancer, there are patients which do not follow the typical course of that subgroup. For example, in a luminal A, which is normally a very good prognosis tumor, there are patients that eventually will relapse with the disease, and so these patients, if they can be stratified in advance, of course, would be eligible, at least in principle, for more aggressive therapy with respect to the bulk of the patient. So essentially, it's the usage of a signature to achieve further stratification in addition to the usual tools which are being used. The entire issue in the field right now is, will we be able to develop stem cell specific drugs? One possibility, of course, is that we will end up getting specific stem cell targets that are only expressed in the stem cells, or they have a good therapeutical delta with respect to the stem cell and the progenitor or other cells. The other possibility is to use old circuitries, which we already know, but we didn't know that were specifically involved in stem cell. One I mentioned is this one relying on NAM and P53, which we think is going to be very, very important. At the end of the day, clearly, stem cell specific therapy might be very important. It's early to be said. In my opinion, it will have to be coupled to conventional chemotherapy no matter what, because there are several ways of making a stem cell. A normal stem cell can derail and give rise to a cancer stem cell, and perhaps that cell would be eligible for stem cell specific therapy. But in cancer, we know for a fact now that even more differentiated cells, like progenitor, can revert to a stem cell state, and they can do it continuously. So the simple idea of sterilizing the tumor with stem cell specific therapy might not be enough, simply because the tumor has found another way to keep on regenerating stem cells, starting from more downstream product. So really, the two things should go hand in hand: debulking the tumor with conventional chemotherapy, and perhaps developing, more specific tool to sterilize the tumors by taking out all the stem cells present at the moment of therapy.